Hello, I'm Steve Cannon, and I'd like to thank the organizers for inviting us to participate in this symposium. Today I'm going to provide an update on our long-term studies of defects of CaV1.1 function in periodic paralysis and how those insights have provided new opportunities for therapeutic intervention.
Periodic paralysis is a rare inherited ion channelopathy of skeletal muscle in which the primary feature is recurrent episodes of weakness that might be quite severe, leaving someone bedridden or unable to even roll over. And these episodes may last for minutes to hours. Another characteristic feature is that these attacks are often brought on by triggers, such as the rest period after exercise, stress, or changes in diet, such as carbohydrate ingestion, potassium, or osmolality. These are autosomal dominantly inherited disorders with classic Mendelian inheritance. Now, all the various forms of periodic paralysis share a final common pathomechanism, which is that during an episode of weakness, the fiber is unable to maintain a normal resting potential of about minus 90 millivolts. And of course, from this sustained plateau depolarization of about minus 50 millivolts, most of the sodium channels are inactivated, and so the fiber is refractory, unable to generate an action potential, and hence there is flaccid weakness. In the presentation today, we're going to be focusing on hypokalemic periodic paralysis. And as the name implies, these episodes of weakness occur in association with low serum potassium, below the normal range from 3.5 to 5 millimolar. So for example, with weakness, somebody might have a serum potassium of 3 or 2.5. These episodes are often triggered by ingestion of carbohydrate-rich meals, which stimulates insulin secretion and then causes a shift of potassium to the intracellular compartment and ends up triggering an attack. Hyperosmolality is also a trigger because this stimulates the NKCC cotransporter, with the end result being a shift in the chloride gradient that promotes depolarization of fibers susceptible to periodic paralysis. This diagram summarizes the missense mutations that have been identified in patients with periodic paralysis. In about 60% of cases, the mutation is in the CaV1.1 calcium channel, and about 15% in the sodium channel, with the remaining being to be determined. Now, amazingly, if you look at these 25 missense mutations, you'll notice 24 out of 25 occur at arginine residues in S4 voltage sensor domains of either the calcium channel or the sodium channel, with this one exception being in S3 of domain 3. This is not a coincidence, of course, because these mutations occurring at homologous sites in both channels all cause the same functional defect, namely the gating pore leakage current. This slide shows one example of the canonical defect that occurs for many of the missense mutations in the S4 segment in hypokalemic periodic paralysis. This was recorded for CaV1.1 expressed in oocytes. This is the R897S missense mutation, which is the outermost arginine in domain 3 of the calcium channel. Cases shown for these voltage clamp recordings are without any form of offset or leak subtraction. And on this brief time scale, there is no significant conduction through the conventional pore of the calcium channel on the time frame of 15 milliseconds over this voltage range. And you can see that for the mutant channel, there's a much larger holding current at minus 100 millivolts. And as we step to voltages from minus 140 to plus 10, there's tremendous inward rectification that occurs in the mutant channel, but not in the wild type. And this is the characteristic signature of the gating pore current, which is permissive when the S4 segment is in the downward conformation at hyperpolarized potentials, and the missense mutation aligns with the hydrophobic waist of the channel. And conversely, in response to depolarization, when the voltage sensor moves in the outward direction, the missense mutation no longer aligns with the hydrophobic waist, and the channel enters a non-conducting state. Over the past several years, a number of laboratories have tested the functional consequences for a number of these missense mutations at arginines in the S4 segment of CaV1.1, which has provided evidence on the consistency with which these missense mutations cause a gating pore current. So of the 10 mutations that have been published, a gating pore current has been identified in 6 cases. In 2 situations, no gating pore current was detected, and 2 others remain to be tested. This shows an example of the signal to noise level required for the detection of the gating pore current. So here for this R900G, which is the second arginine in S3, you can still see a gating pore current, an excessive inward current that's greater than wild type. And this is more typical, that the magnitude of the current is twice the background nonspecific leakage current. The first example I showed you was a rather spectacular case of the largest gating pore current that's ever been seen for this R897S. So a more typical is the R900G, and in comparison, we were unable to convince ourselves that there was an anomalous gating pore leakage current for the R900S. So at the same residue, but serine instead of glycine, because the current voltage relationship was not distinctly different from that of wild type. Because no gating pore current was detected for some of the HypoP mutants, this raises the question whether other functional defects of CaV1.1 might be responsible for the dysfunction of the muscle fiber. The natural question would be, what about voltage-dependent calcium release? Of course, we'd like to test this for these two missense mutations in domain 3, for which it was not possible to detect a gating pore current. But the available tool at this time is the R528H knock-in missense mutation in the mouse. This is the most common cause of hypokalemic periodic paralysis. And while it is not one of the mutations without a gating pore current, at least it provides some index of whether voltage-dependent calcium release is impacted by these missense mutations in the voltage sensors of domain 2 or 3, which of course are adjacent to the critical 2-3 intracellular loop for excitation-contraction coupling. To address this question, we recorded the calcium transients with the fluorescence change in Oregon green in response to action potentials that were triggered under current clamp. So we used a holding current so that the wild type and HypoPP fibers in our homozygous R528H mouse were both starting from a resting potential of minus 85 millivolts. And you'll see the amplitude of the action potential was the same, but the peak of the calcium release was lower for the R528H fiber. And this shows data from about 20 some odd fibers, either from wild type or R528H. And you could see there was a modest but consistent reduction in peak calcium release for the HypoPP fibers compared to wild type. The release transients were also measured under voltage clamp protocol so that we could more precisely measure the voltage dependence of release. And here you see the waveforms for wild type compared to the homozygous mutant fiber. And again, we see essentially the same defect, which is the peak of the calcium transient is lower in the homozygous R528H mouse. And here is the voltage dependence of the peak, showing that there's no difference in the steepness or the midpoint. Instead, there's about a 20% decrease in the peak calcium transient revealed under voltage clamp. So because the defects in calcium release were quite modest, it's not thought that those would be sufficient to cause the severe symptoms in the patient. So we're going to return to the primary problem, which is maintaining the resting potential, and think about how the presence of that gating pore leak influences stability of the resting potential. This just schematically shows a model to simulate the resting potential of skeletal muscle fiber. So we include a number of voltage-dependent conductances, the inward rectifier potassium channel, as well as a delayed rectifier, the CLC1 chloride channel, the sodium-potassium ATPase, as well as this NKCC cotransporter. Even with this somewhat reductionist model, if you look at the predicted resting potential as a function of extracellular potassium, you'll see it initially follows sort of the Goldman-Hodgkin-Katz type of prediction with hyperpolarization as the extracellular potassium is reduced. But then all of a sudden, a critical level is reached, at which point even wild-type fibers will paradoxically depolarize. And this is because in extremely low potassium, lower than would ever be seen in vivo, the inward rectifier conductance fails, and the fiber depolarizes until the delayed rectifier can be sufficiently activated to prevent further depolarization. So the defect in the presence of the gating pore leakage current in hypokalemic periodic paralysis, regardless of whether that leak is from the NaV1.4 sodium channel or the CaV1.1 calcium channel, in either case, this leakage current causes a shift of this relationship between extracellular potassium and resting potential. And so with this right shifted curve, now as the potassium is reduced somewhat within the low physiologic range, there's the possibility of this paradoxical depolarization in low potassium for fibers with hypokalemic periodic paralysis with the gating pore leak. And of course, from this depolarized resting potential, these fibers will be inexcitable because a substantial portion of sodium channels will be inactivated. Recently, we've been able to corroborate this theoretical prediction of paradoxical depolarization in low K with continuous recording of the resting potential in mouse muscle fibers in vitro. So in the left panel here, you're just watching a very long 2-hour continuous recording of the resting potential with a sharp microelectrode, showing what we just showed in the model, which is that, starting with a normal potassium of 4.7 and a resting potential about -78, when the potassium is lowered as predicted from the leftward shift in the Nernst potential for potassium, a wild-type fiber hyperpolarizes. If you go back to normal potassium, you return to the prior resting potential. Even at 1.5 millimolar potassium, there's a hyperpolarization. But if an extremely low potassium challenge is applied, even to a wild-type muscle, what starts out as hyperpolarization paradoxically depolarizes to this plateau potential. And this is exactly what you saw in the prediction when there's failure of the inward rectifier. This is reversible such that if the extracellular potassium is once again returned to 4.7 millimolar, this fiber will repolarize close to the original resting potential, and it's repeated again with an even lower challenge at 0.5 millimolar potassium. In the panel on the right, we're showing the response from one of our mouse models with hypokalemic periodic paralysis. This is one of our most spectacular examples. It happens to come from the mouse with a sodium channel mutation. I'll show you this also happens for the calcium channel mutation. But here you can see starting at a pretty decent resting potential of about -68 millivolts. And when the potassium is lowered to 3 millimolar, not very low at all, there's a paradoxical depolarization. Similarly with 2.5 or 2 millimolar. So ion concentrations that would have caused hyperpolarization in a wild-type fiber causes paradoxical depolarization in a fiber susceptible to hypokalemic periodic paralysis. As promised here, I'm showing an example from the R528 homozygous calcium channel mutant mouse. You can see again, when the potassium is decreased from 4.5 to only 3 millimolar, there's a paradoxical depolarization. And in a muscle from a different R528H mouse, a decrease to 2 millimolar potassium also caused paradoxical depolarization. So the paradoxical depolarization of the resting potential highlights an obvious therapeutic strategy, which would be to somehow try to stable the resting potential, especially in low normal potassium levels. Opening a potassium channel conductance would favor that hyperpolarization. In the study I'm about to show, we looked at an opener of the CaV7 family of potassium channels with retigabine. We used isometric force as the outcome measure to test for the effectiveness of the K-channel opener retigabine. These traces are showing isometric tetanic contractions from the R528H homozygous HypoPP mouse. Here you can see in normal 4.7 millimolar potassium, the peak force is 13 grams, and in the presence of a 2 millimolar potassium challenge, the force in the soleus decreases about 25% to 10 grams. This muscle from the other limb in the same animal was tested in a dish that was exposed to 10 micromolar retigabine. And you can see in the presence of the drug, the control response in 4.7 millimolar potassium is within the normal range. Most importantly, with a 2 millimolar potassium channel, there's no loss of force in the presence of 10 millimolar retigabine. Retigabine is also effective at preventing the attacks of low K-induced weakness for the sodium channel mutant mouse. We happen to have a full dose response curve available in that animal. You can see again, starting at 10 micromolar completely protects the muscle from a loss of force with a 2 millimolar potassium challenge. Substantial protection occurs even with the low micromolar range. Importantly, dose response curve is about 30 times more potent than was available from another potassium channel opener, panacedyl, which promotes the opening of KHP channels. The dosage of panacedyl required to provide substantial protection is about an order of magnitude higher than can be achieved in patients. And secondly, attempts to use pinacidil have caused limiting drop in blood pressure and risk of fainting, whereas retigabine, which was initially marketed as an anti-epileptic medication, is a viable alternative to stabilize the resting potential and reduce the risk of transient attacks of weakness and hypokalemic periodic paralysis. The other approach for management of inherited channelopathies, of course, is gene editing to either remove the mutant allele or correct it. We started to look at these approaches in periodic paralysis because while the abnormalities in resting potential and fiber excitability provide a lot of strategies for how to prevent the transient attack of weakness, these patients also get fixed permanent weakness that's slowly progressive in age 40 and 50, for which the mechanism is unknown and is not clearly related to fiber excitability. And so if we're going to make any progress in reducing the risk of permanent weakness, gene editing is the best approach towards that. So we've used two methods. The first is to disrupt the mutant allele in the R528H HypoPP mouse. Disruption of the mutant allele is feasible for patients with hypokalemic periodic paralysis because all of them are heterozygotes, so they have one normal and one mutant allele. Muscle fiber remains viable with a single functional copy of the CaV1.1 calcium channel, as we all know from the muscular dysgenesis mouse. There have been cohorts of studies in the families of patients with muscle disease in which asymptomatic relatives are carriers of null mutations in CaV1.1 and have no muscle dysfunction at all. The other favorable aspect of disrupting the mutant allele is that the nuclease efficiency of CRISPR-Cas9 is highly efficient, so you're likely to be able to disrupt a substantial fraction of the mutant allele. And finally, computational studies show that disruption for even a fraction of the mutant allele is probably sufficient to cause meaningful improvement in muscle function. So while gene editing with CRISPR-Cas9 is now becoming routine, as any of you know who've tried this, there are a lot of conditions that need to be met for your specific circumstances. And so we were working to establish proof of principle for HypoPP mutations in CaV1.1. The task before us is that the guide RNA will be different from wild type by only a single nucleotide, and there needs to be a protospacer associated motif close by such that the guide RNA Cas9 complex will recognize the site for cleavage. And then this double-strand break is followed by random insertions and deletions, so-called indels, that will cause spurious frame shift and disrupt the function of the gene. Our protocol was to start with the homozygous R528H mouse, establish myoblasts from neonatal muscle, then transfect with the Cas9 endonuclease and guide RNA specific for the mutant allele of CaV1.1 that's coding for the R528H. We then FACS sort on a GFP reporter to collect the successfully transfected cells, and then analyze the genomic DNA by Sanger sequencing and use deconvolution algorithms to detect the number of insertions and deletions. So here's an example for in vitro editing with CRISPR-Cas9. So this is the chromatogram of the Sanger sequence in the region of the mutation for R528H, and highlighted in blue is the segment of sequence to which the guide RNA was directed. What you can see is that the chromatogram is clean for the wild type, indicating there was no double-strand break and random insertions and deletions occurring here. But for the mutant allele, using these very same constructs, in the region of guide RNA, there was a double-strand break and these random insertions and deletions such that the Sanger sequencing now had a heterogeneous template. You see a lot of garbage and spurious peaks here, which when deconvolved indicates that about 60% of the mutant allele was disrupted for this in vitro transfection. So clearly showing sufficient efficiency and specificity for this CRISPR-Cas9 approach to delete the mutant allele. In further studies, we've also shown this gene editing is possible in vivo. So we electroporated the muscles of the hindfoot in the mouse. Here you can see the reporter expressed in the FDB and interosseous fibers, which was also electroporated with our guide RNA and Cas9 construct. We then dissociated these fibers so that we could isolate GF-positive individual fibers, isolate genomic DNA, and look for evidence of indel formation. And again, you see just downstream from the guide RNA the Sanger sequencing becomes a complete mess because of all these indels. And in vivo, we achieved almost 50% gene editing, again, in a proof of principle showing it's possible to edit the CaV1.1 gene in vivo. The alternative gene editing approach is, instead of disrupting and destroying the mutant allele, to correct it with adenine base editors. This, of course, has the advantages of there's no double-strand break created by Cas9 nuclease. The transcript level is preserved. You're not going to create haploinsufficiency by deleting the mutant allele in a heterozygous patient. And also, similar to removing the deleterious allele, if you look at the effects of correction, the model prediction is that a modest amount of correction is likely to be sufficient to cause a significant improvement in muscle function. So we performed pilot proof of principle studies. For these base editor constructs, we had to use a relaxed PAM site with an NGN motif. It wasn't possible to use the canonical NGG. It's a dead Cas9, so there's no endonuclease activity merged with the adenine base editor. And thus far, we've done this with the same in vitro assay, again, isolating myoblasts from our homozygous mutant mouse, this time transfecting with all the machinery for the guide RNA and the base editor, along with the GFP reporter, sorting on the successfully transfected myoblasts, and then Sanger sequencing and looking for evidence of editing. So here you can see an example of the chromatogram from our sequencing. Here is the missense mutation that causes an arginine to histidine. The mutant has adenine, which is supposed to be guanine, and you can see with this edit machinery, we were able to convert 34% back to the wild type. So this is typical for the level of efficiency that can be achieved with editing as opposed to nuclease and indel formation. We're still going to try to optimize this further and verify this occurs with in vivo electroporation. And our next goals are to scale this up with adenovirus transfections and look for change in susceptibility to loss of force in the muscle as our outcome measure of the efficacy of gene editing. So to summarize, it's still a consistent story that the overwhelming majority of missense mutations in hypokalemic periodic paralysis are missense mutations at arginines in S4 segments. Most of those cause a gating pore leakage current, and there's only a mild disruption of calcium release, at least for the mutation that has been studied in S4 of domain 2. This information has provided a number of strategic approaches. The first is to counteract the consequence of the gating pore current by stabilizing the resting potential through the usage of potassium channel openers. We've also done some preliminary proof of principle studies showing that selective and specific gene editing of the mutant allele can be performed for CaV1.1 based HypoK, either to destroy the mutant allele or correct it with base editors. And so finally, I'd just like to end by thanking the very talented people who performed these studies. Fen Wu, the scientist in the lab who did all of the gene editing experiments, as well as those cut open voltage clamp experiments showing the gating pore current. Marino DeFranco made all of the calcium release measurements, and Marbella Canones maintains our mouse colony and also performs the isometric twitch studies, and here are our funders. So thank you very much. It was wonderful to be able to participate in this symposium.